Genetic disease represents an immense healthcare burden, affecting 1 in 10 Canadians. Our DNA is made up of billions of letters that can be strung into sentences known as genes. Genes, in turn, are responsible for every single function that occurs in our body. Now, 30% of all genetic diseases are caused by just one single typo in one of our genes. This typo often represents a death sentence. But what if it didn't have to? What if we could correct this typo? Well, thanks to a new technology called a base editor, we actually can. These are proteins that can be directed to a specific site within our DNA where they recognize typos and convert them back to a healthy letter. But since different genetic diseases affect different tissues, we need to make sure that we are accurately able to direct the base editor to diseased tissues. Currently, this represents a major barrier for the clinical use of gene editing. My project focuses on expanding the range of tissues and therefore diseases that we can use editors on. To do this, my lab and I have developed a mouse model that enables me to tell exactly where I've edited. We've taken the gene that fireflies use to glow, and we've added that to a mouse. But we've also added a single typo. This means that when I add a base editor to my mouse, any of the tissues that I have edited will now glow. The ones I haven't will not. I can use this to screen the different delivery methods. I currently use lipid nanoparticles to package and protect my base editors. These are tiny bubbles of fat. They are made up of 4 different lipid components. I can change each of these components to alter where in the body my particles are going. Let's take the example of targeting the brain. To do this, I attach a neurotransmitter to one of these lipids. The body recognizes that neurotransmitter and then brings it to the brain. After this, I can make changes to the shape and to the properties of the lipids themselves. Anytime I make a positive change, I will see that the brain of that mouse will glow even brighter. I can then apply the same strategy to different organs, hitchhiking on the body's pre-existing transport pathways. This enables me to perfect gene editing in multiple organ types, bringing us one step closer to being able to treat that 30% of genetic diseases that currently have no treatment. And it all starts with one glowing mouse. Thank you.